Thank you for joining. I will present some of my postdoctoral work that I've done in University of Liège in the Physiology of Cognition lab. And I was supported for this by the FNRS. And it will be talking about cardiac responses to sounds and what we can learn from these cardiac responses about sleep and the embodiment of sleep functions. So I don't know how much you are familiar with sleep. So sleep is classically defined as a state of behavioral unresponsiveness, but it doesn't mean that nothing happens whenever we are asleep. And you know that we have a lot of intense cognitive activity from remembering dreams, for example, in the morning. And lots of the research about sleep has benefited from neuroimaging because despite the lack of access to behavior or limited access, at least, we can look at what kind of variation in terms of brain signals we can observe during sleep. And what has been discovered is that actually sleep is the host of a lot of different types of activities. And one of the largest actually electroencephalographic deflection that we can observe is occurring during sleep, and it's called K complexes, which are these large deflection in a stage that we here is named N2, which means light non-REM sleep. And whenever it's happening in isolation, it's called K complexes. But whenever it's happening in trains, it's called slow waves. Actually, if you have more than 20% of this activity in your EEG signals, it means you entered rather deep non-REM sleep. It has also been discovered another sleep stage, which notoriously the EEG activity looks a lot like wake activity, meaning really desynchronized. And there is another hallmark that we can observe during REM sleep, which are these ocular movements, which gave the name of rapid eye movement sleep. And interestingly, rapid eye movement is not happening 100% of the time during REM sleep. Actually, the majority of REM sleep is devoid of ocular activity, and it's called tonic REM. And 20% is these rapid eye movements happening in bursts, and it's called phasic REM. So this is more a general introduction into the physiology of sleep at the brain level. And why is it really useful to use neuroimaging is that classically psychology has relied upon behavioral responses during sleep to obtain behavioral responses during wakefulness, and this is how we gain some insight into cognitive activity that have been happening during wakefulness. But with sleep, we have limited access because the motor output is largely diminished, as you can see here in the EMG level. And so we can use the brain signals actually to gain insight into the cognitive activity. And this has been the focus of my PhD work in École Normale Supérieure in Paris, where we use decoding technique from brain signals to probe cognitive activity during sleep. And the idea is that whenever we sleep, we disconnect relatively from the environment. So we can supposedly make internal processing like memory consolidation, dreams, etc. But we still able to process external stimuli to some extent, which allows us to wake up, for example, whenever there is an external threat, or whenever we hear our alarm clock. So we investigate this balance between the ability to focus inwards on our own activity or to be able to watch out for external stimuli. And so we use for that a classical paradigm called the cocktail party, whenever we have in one ear some French meaningful speech, and the other ear we have Jabberwocky. And we used a EEG decoder, which tracks the variation of speech sounds to be able to then identify which of the two streams is preferably amplified in brain signals and thus attended. So people were instructed to focus on the French speech stream during wakefulness over the meaningless speech. So Jabberwocky is actually a a speech composed of pseudo words. So each meaningful word has been replaced by some words that sounding French, but is actually meaningless. And so people are able to selectively amplify the French speech during wakefulness. And here Guillaume Legendre found in this study that during light non-REM sleep, you're still able to selectively amplify the French speech, but in deep sleep, you are not able to, or if something, you are even selectively a bit suppressing the French meaningful speech, especially so during slow waves. So these large deflections that I've been showing you before. And I carried out the same paradigm during REM sleep in morning naps and showed that actually during REM sleep, most of the time you are able to selectively amplify the meaningful information. And during isolated eye movements, you selectively actually suppress the French speech. And if you draw on this distinction between tonic and phasic REM, you see that there is selective amplification of meaningful information during tonic REM, and which is lost during phasic REM. So there is a striking parallel between non-REM sleep and the division between light and deep sleep, and whatever happening during REM sleep between tonic REM and phasic REM, despite having different physiology. So it shows that actually, to some extent, we are able to flexibly attend to the external sounds, even if we are sleeping, and we can do so to favor external meaningful information or to selectively suppress them whenever it might interfere with our internal activity, because we know that ocular movements are associated preferably with intense dreaming activity and slow wave with memory consolidation processes, for example. We also investigated to some if we can learn some new information during sleep. And here we presented some Japanese words during sleep with barking sounds of a dog for the word ine, meaning dog in Japanese, in naive participants. And we showed in a test in the morning that people were able to recognize which of the two images referred to the Japanese words. So here the image of a dog for ine, and they were able to do so for stimuli that were presented in non-REM sleep. And we analyzed the responses at the brain level of whenever these stimuli were presented and identified some frontal slow wave response that was predictive of the fact that people were able to selectively encode this information during non-REM. So basically here in my PhD, I provided evidence that the brain is actually processing information to a large extent during sleep, both able to selectively attend some information in a flexible way, or also encode some information. And this also depended on sleep stages and the microphysiological events like slow waves and ocular eye movements that were associated. But one thing that was left out of the picture in this work is what about the body? And actually we know that the body bodily activity is also undergoing some significant variations across the wake-sleep cycle. So if you look here for example, to the cardiac activity, you have a slowing down of the cardiac activity as indexed here by the heart rate, which is lower during non-REM. So here it's called S1, S2, and S3. And whenever you enter to a REM sleep, you have a bit more elevated heart rate. So there is this change in terms of heart rate over the different sleep stages. And what about cardiac responses to sounds? Actually, the literature is very limited in investigating this question. So we know for a long time that if you play a sound during sleep, like a basic tone, you can elicit a large K complex here. Actually, for the anecdote, K is called K complex because it's referring to the fact that some doctors were knocking on the door. So whenever they looked at these signals, they indexed like a K for knock. And if we look at the cardiac activity, we see that there is an elevation of the heart rate. And this is basically, to the extent of my knowledge, the only kind of evidence that our research that have been done on cardiac responses to sounds during sleep, and nothing has been done on more complex sounds. So basically, that was the focus of my postdoc, to approach this question and to fill this gap. So do you have any questions so far, or is it clear? Okay. So the question that I've been addressing is whether cardiac responses to more complex sounds can inform us about cognitive processing during sleep. And to give you a bit more motivation why I approach this question in this way is maybe we can refer to a framework which is called the 4E. So the 4E is the idea that cognition is embodied first, so meaning that the way that we think is also involving our body. It's extended because also the tools that we are used to work with is also shaping the way that we think. It's embedded because the thinking that we have is also included depending on the context in which we are embedded, and there is constraints by the context about which type of cognition we will do. And it's enactive, meaning that there is strong link between action and perception. And this 4E, sometimes there is last E that is added, which is ecological, because this all refers to the fact that our cognition is actually not just the brain in a vat, but it's in an environmental context, and it's by the interaction between the brain and the environment using our body that cognition emerges. And I tried to develop an approach of something like an ecological science, not just in terms of the philosophical framework, but also the way I'm doing science. And if you're familiar with the 5R in ecology, is referring to refuse, reduce, reuse, repair, recycle. And the first thing I did is to think that there is a lot already of sleep data that have been acquired in the literature. And rather than going for more experiments, maybe we can think in terms of resources and the ecological footprints of our scientific activities, and refuse to go into more experiments, reducing then the amount of maybe new data being produced, and reusing actually already existing data sets that have been investigating sleep cognition but have neglected or left out the analysis of bodily signal, and in particular ECG activity. And we can do so by gathering large data sets. We can also take a second look at some of the claims that have been done in the literature, and maybe repairing some kind of outlook that we have on what is the cognition during sleep in the literature, and recycling some of the tools that have been used in the past. And I've been linking this with the open science framework that have been in a recent presentation to the Open Science Day that you can check out if you are more interested. So this approach is very much compatible with the open science mindsets by reusing existing data sets, promoting collaborative practices over competitive ones, and sharing resources. And so the work that I will present to you is actually a reanalysis or a secondary analysis of ECG activity from some pre-existing data sets. And the first study I will talk you in a minute is this study by Jonas Beck, Erna Loretz, and Björn Rasch. So it's a Swiss team that investigated what happens if we present some relaxing words during sleep. And what they found is actually when you present some relaxing words, you will sleep deeper and with a better sleep quality. And they had this hypothesis that, and they compared that to neutral control words in 2 different nights. And they had 50 subjects in this included, and they analyzed the EEG activity to make this claim, because they looked at the amount of slow wave sleep, but they haven't investigated ECG. And their claim was actually that the perception during sleep is here at the semantic level, so meaning that we can understand that which words are relaxing, refer to the relaxing meaning rather than the neutral one. And but more than that, we just not process intellectually these words, but we also experience them. And that's why we sleep deeper and better if we are hearing relaxing words. And there is an embodied processing of the words, meaning that this will affect the physiology of sleep to hear some relaxing words. And this opens non-pharmacological, non-pharmaceutical ways of improving sleep quality. So this is the first data set that I will present you, the cardiac analysis, because I wanted to carry out this question about whether we can find evidence in the body of this relaxation. The second study is gathering 2 different data sets that use the same paradigm, which is a very influential paradigm called the local global paradigm. And it's a paradigm used to look at hierarchical novelty detection. In here 56 subjects, and both have been looking at this paradigm at the brain level, but they haven't investigated the cardiac level. And across both study, I will actually reuse a methodology that have been designed by Federico Raimondo and colleagues, where they looked at the local global paradigm in non-communicative states, and more precisely, disorders of consciousness. So disorders of consciousness is the states when you go out of the coma, and you have you cannot communicate, but you can look at the brain signal to differentiate between if you are in vegetative state or minimally conscious states. And they looked actually at the cardiac activity. And to do that, they extracted it from actually what is considered classically an artifact in the EEG, because you can capture some of the heartbeats from the EEG, and usually it's discarded and thrown away because it's considered as noise. But here it's been used to look at what happens whenever you present words, some sounds. Here it's so in the local global paradigm is here, for example, if you have beep, 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 bop. So even the fifth sound is a deviant compared to the previous ones, and you can look if the next heartbeat here, which is the R peak, is coming a bit faster or a bit slower as compared to the distance, the duration separating the fifth sound and the preceding heartbeat. And this is the methodology I've been using across both experiments. So this methodology, I've been also like developing tutorials so people can check it out in an open science way if they want to reuse it, and with some introductory step in how to apply it to your data set. Here again, if you have any question, please let me know if you need some clarification at this point, and we'll enter the results, presentation of the results. Is it good?
I have a question.
Sure.
So you're using a kind of oddball stimulus. I didn't quite understand. So are you saying the heart rate reacts to the oddball stimulus as well? Like a kind of
Sure.
startle or a surprise sort of thing? Or yeah.
So what they found in so there is two different kind of oddballs. I will present it in the study too, and we can come back to the results they found also in disorder of consciousness if you want to compare. But basically, the idea is that you have two kinds of oddballs. So you have the beep 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 bob. So this is a local mismatch because it's the last sound is different from the four preceding ones. And you can have also a global deviance, which is if you have like beep 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 bob, beep 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 bob, beep 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 bob, beep 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 beep. So here the very last sound is the same as the preceding ones, but the sequence is different from the four preceding ones. So you need to integrate information over a longer period of time to be able to detect this kind of novelty. And this is supposedly associated with conscious processing because it has been used to differentiate different levels of consciousness when you emerge out of the coma state. And the more you have like a sign of this global deviance, the more conscious you are, so to say. So it has been applied then to sleep to see whether you can track also variations in terms of different consciousness levels, or if you are a bit less ambitious in your claims in terms of novelty detection, to see if we have we can still carry out complex information processing during sleep. So that's what I did. I will present this in study 2. First, I will start with the study 1. And the study 1 is about whether we can, the cardiac responses to relaxing world can reveal the embodied nature of semantic processing in non-REM sleep. So this is the paradigm in detail. So they had participants undergo 2 nights, and one which is presenting relaxing cues. Here, for example, the word relax. And there is also a control which is the reverse. So here it's called reverse relax. So it's basically the same words, but in reverse to check for acoustic responses and to see if there is really a specific processing of the meaningful information here. And there is the same thing for neutral words like produce or materials that is presented in a different night. And so what they found in this paper is that you have an elevation in terms of slow wave sleep, so the deeper non-REM sleep stage, which is correlating with the subjective sleep quality. So what we did is to use this methodology I presented it to you. So we compared the difference here between the duration between the heartbeats and the presentation, the onset of the words coming after the stimulation, and we compared it to the one before. We did the subtraction, and then we will compare whatever happens for relaxing words compared to their reversed version, and for neutral words compared to the reversed version. And what we found is actually for the relaxing words, there is a slowing down of the cardiac activity, because here there is the heartbeat is coming a bit after in comparison to the heartbeat before, whenever you subtract it to the reversed words, just to check for having acoustic control. And this difference is not found in the control condition. And so to make this point a bit clearer, we also look at overall during the presentation of the cues during the night. We can see that whenever we present the cues during non-REM sleep, the interbeat interval, which is an index of the heart rate, is a bit bigger. So meaning that there is more distance between two heartbeats whenever we present the cues during the relaxing nights compared to whenever the cues are not presented. And this effect is not present again during control nights. So we have really a slowing down of cardiac activity, which is happening whenever specifically we present the relaxing cues during night. And this is coming right after the presentation of the words. So it's confirming that this is the processing of the words that is tightly linked to the slowing down of cardiac activity. And the last thing we wanted to do is also to compare with the EEG results, because from the experiments, whenever they analyzed also the EEG, and they found some specific markers in terms of slow wave activity that was enhanced during relaxing nights as compared to control nights. And so we put both the brain markers and the cardiac markers in a model to try to predict the difference between relaxed and control night. And we saw that both ECG and EEG predict some of the variance that is differentiating relaxing and control night. And if we add both together, we outperform the models using brain activity alone or ECG activity alone. So it means that actually there is additional information in the ECG as compared to brain activity alone. So this shows that actually taking into account the cardiac activity whenever you look at the effect of the presentation of relaxing words during sleep is actually gives you some extra information in the investigation of the difference between relax and control night. So this is the conclusion of the study one, that you can observe a cardiac deceleration for relaxing versus reversed version of the words only for this condition and not for control condition. This is confirmed that there is a cardiac deceleration overall during the presentation of the relaxing words. And these effects are at least partially independent from EEG activity. So this is a motivation for looking at cardiac activity, because it holds some important information. So do you need any clarification to this stage, or is it understandable? So now we'll jump to the second study. And so here we look at whether cardiac response to deviance, as I presented to you in the local global paradigm, can reveal the variations of hierarchical novelty detection during sleep. So as discussed and presented before, so there is different, this paradigm is using different trials. So there is trials that are just the sequence of 5 similar tones like beep, 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 beep. So they are called standards. And there are some sequences that have a deviant like beep, 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 bop, and they are called deviants. And actually, these deviants can be local. So if you have beep, 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 bop, repeat, sorry, these deviants can be global standards. If you have these beep, 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 bop sequences that are the mostly occurring within a block. And in another block, these deviants are more global deviants. If you have usually beep, 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 bop. So here it's both global deviant and local deviant. So it's a bit integrate, but the way that these local mismatch effects are computed in the literature classically is by looking at the local mismatch effects by summing the local deviants, which are here the beep, 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 bop, and comparing to the local standards, which are beep, 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 beep. And you can compare, and there is also the global deviance effect, which is, for example, here the beep, 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 which are in the sequence where it's the minority of beep, 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 beep within the block. And you have you sum it with the global deviance, as mentioned before, and you subtract it to the global standards. And so by using this computation, Melanie Strauss and colleagues in this seminal study, they looked at the variation of these brain responses at the brain level during sleep. And they found that actually you have a preservation of a local mismatch effect during sleep, both in non-REM 2 and REM sleep. And you have a loss of global deviance effects in non-REM 2 and REM sleep. And they argue that actually sleep is disrupting the processing of global information in the brain. And so this is a sign, at least this is their interpretation, that you have predictive coding machine that is operating during wakefulness that is not as much functional, at least for the complex information during sleep. So what I did is to look at what happens to the cardiac responses. And so I compared also here the pre and the post duration, and at the local mismatch mismatch level, surprisingly, we found no difference in terms of responses. And when we looked at the global deviance effect, we found one in REM sleep. So it's very much at odds with the brain results. And when we look more in detail by drawing this distinction between the period of the REM sleep when there is no rapid eye movement. So we, in the introduction, I presented you how we have amplified processing of external information in this state. We have the presence of this global deviance effect, which is lost in phasic REM. So basically what we show here is that contrary to the prediction that we can draw based on the analysis of brain signals, the analysis of cardiac activity offers you a strikingly different picture and demonstrate that there is actually hierarchical processing of complex information during sleep. So these results have been published as a preprint, are now under review, and you can check them out if you want. And these are the results we obtained when we used the same parameter that Federico Raimondo used for DOC patients. So these are what we had as a confirmatory analysis. But whenever you look into sleep, actually sleep is the cardiac activity is a bit slower than during wakefulness, as I presented you before. So if we change a bit the parameters into and the window on which we look at cardiac signals, I don't really enter the technical details of it. We also found that actually we can uncover some local mismatch effects in non-REM sleep exclusively, which are also found in light non-REM sleep, and we can and using this larger window, we don't find any global deviance effects. So there is like a slight dependency on the parameters that we use to undergo this analysis. So these are more exploratory results. And this still, it's not like it's a very new field. So there is a lot of open questions. But I will just first, before going into discussing the interpretation of these results, I will just briefly summarize these results. So what we observe here is a deceleration to global deviance effects in REM sleep, an acceleration to local mismatch in non-REM sleep, and we found that we have a variation of these responses depending on the presence of the sleep hallmarks, meaning rapid eye movements, and maybe also the difference between light and deep non-REM sleep based on the presence of slow waves. So it reveals some of the variations of hierarchical complex information processing and novelty detection throughout sleep stages. So was this clear for you? This one is a bit more technical in terms of the way that the effects are computed. Yes, Alice? Oh, it's good? Okay. So how do we interpret these results? So if we take one of the framework which has been put forward to interpret the variations of cardiac activity during wakefulness is based on the interplay between sympathetic and parasympathetic activity. And the idea is whenever we perceive some threat or some error, usually what we observe is a deceleration of cardiac activity. And the idea is that by decelerating a cardiac activity, we actually have, we enhance perception abilities. So we can analyze whatever is going on during in the perceptual scene. And then the parasympathetic brake is removed. And so we have sympathetic activity coming back that is more tuned toward action. So elevation of cardiac activity that will allow us to react. So if we compare it to whatever we found during sleep, is that we have a slowdown to relaxing words and to global deviance. So here the idea is that, and we have a speed up or an acceleration to local mismatch. So it's a bit more complex picture than this simple picture that has been offered during wakefulness. And it's may depend also on the meaningful and the meaning and the semantic content of the perception of the words perceived. And depending on whether it's like an error at a high level, it's so be tuned toward perceptually processing the information, whenever it's a more local mismatch, which is more triggering an automatic arousal response. We are more like directly tuned to our action and react without like much more perceptual processing and thinking. And might explain why we have more like acceleration of cardiac activity. So another way to look at it is to look at the variations of the level on which we have heart-brain interaction during sleep being prevailing. So in wakefulness, we have a balance of both low-level and high-level control of cardiac activity. And actually, frontal activity is rather inhibiting cardiac responses, and that may explain why we also don't observe so much cardiac responses during wakefulness in the local global paradigm. And this is also something that was found in the previous study by Federico Raimondo, so it's kind of a replication and robust effect. But in sleep, we know that frontal activity is reduced, and so that might uncover a regulation of cardiac responses by more like low-level brainstem regions, which are more tuned towards arousal and reactivity during sleep, and more maybe complex high-level processing during REM sleep that may explain why we have more like global deviance effect. And actually, we also, so it's a bit a simplistic here again picture, because we found that we can also have this kind of response to relaxing words during non-REM sleep that are more like high-level type of processing. And it's still unknown whether it's because we have a general alteration of sleep patterns, like the triggering of slow waves that are also linked to arousal, and sleep deepening that may be mediating the cardiac slowdown to relaxing words. And also in REM sleep, if we focus on tonic REM, like I haven't shown you those results, but in some kind of local mismatch, some trial, we also find an acceleration in tonic REM to some of the local mismatch, as we've observed in non-REM sleep. So maybe there is also this kind of level of arousal, low-level responses during REM sleep. So this is maybe another simplified framework, and investigating all these cardiac responses to sounds during sleep might pave the way to have a more complex and integrated picture about what is the level of the interaction between cerebral activity and cardiac activity during sleep. So as I explained to you, there is like relatively few studies that have been taking into account cardiac activity during sleep. So here it's one of the first models that emerged in 2022 by Whitehurst and colleagues that offer the role for autonomic activity into memory consolidation processes during sleep. So you can check it out if you are more interested in the subject. And there is like a whole model showing the interaction that can be found in between cortex and arousal brain regions. And interestingly, they also like showed that if you take into account the variation of cardiac activity during sleep, you can compare the part of variance explained as compared to using just brain activity alone. You can explain more of the variance in terms of memory consolidation effect, and showing that there is really a part of the autonomic activity that is very important to take into account to understand memory benefits happening overnight. But luckily, there is like another study that showed that actually using this targeted memory reactivation during rapid eye movement, you can promote the reduction of emotional memories. And they found like how it's, for example, the response at the brain level and the cardiac level with these emotional memories during sleep. But they didn't analyze for ECG what's again during sleep. So this is something that is left to be done. And I hope like with all this postdoc work to motivate a wide range of researchers that cardiac responses to auditory information are worthy to acquire and include in analysis. They are informative for the study of sleep function, and there are many new questions arising from all these studies that are worthy to investigate. And they are complementary to brain activity whenever we want to understand the sleep functions. And one motivation for that is that for cognition might be actually embodied in the sense that we cannot just focus on brain activity to understand cognition, and we need also to include bodily signals. And so the take-home message is that cardiac responses to auditory information can reveal some unique and embodied aspects of cognitive processing in sleep. And this might be generalized maybe to all states where we lack direct communication with the people and on which we are investigating cognition. So it can range from disorder of consciousness patients, where we have limited ability to communicate with them, but it can go also to anesthesia, and it might be important to understand what type of experience people live whenever they are in the operation room, for example, and whether there is a bodily responses that can reflect some kind of pain processing, for example, or even semantic information processing. And this can also extend to psychedelic states, for which we know that there is difficulty of sometimes verbalizing or getting some behavior. And we can look at cardiac activity it has been started to be done to really gain insights into these conscious states. So I want to thank the team I was part of during my postdoc that was supportive in these projects, and also the collaborators that were involved in both of the studies. And thus I'm open to any questions about these studies and this work. Thank you.